Hi, everyone. Welcome to today's webinar. My name is Catherine Bryant, and I'm the founder of the Speak Foundation. Today, we are presenting a great webinar on outcome measures. What are they, and why are they important to clinical trials? In just a moment, I will formally introduce Dr. Alfano, but I want to share that she is our speaker today. She is a principal investigator and assistant professor. She works at the Abigail Wexner Research Institute at Nationwide Children's Hospital Center for Gene Therapy. You know, this is an exciting webinar because this is a topic that a lot of individuals with limb-girdle want to learn about, especially because clinical trials for limb-girdle are going on right now. We want to thank Sarepta Therapeutics for sponsoring our webinar series. And Sarepta Therapeutics has brought together the essential elements for development of gene therapies: productive collaborations, unparalleled scientific understanding, and an unwavering sense of purpose. Sarepta has 5 limb-girdle gene therapy programs in development, including for subtypes 2E, 2D, 2C, 2B, and 2L, and 2A in collaboration with Nationwide Children's Hospital. Sarepta welcomes the opportunity to connect with the limb-girdle muscular dystrophy community. You can meet Sarepta at emailing advocacy@sarepta.com. You can stay connected by going to limb-girdle.com/stay connected, or you can follow Sarepta. They're available on several social media platforms. And as well, we want to let you know about the International Limb-Girdle Muscular Dystrophy Conference that's coming up later this year from September 17th through the 20th. And this is a virtual event. And our keynote speaker is Dr. Scott Gottlieb, who is the former FDA commissioner. He will be speaking to you as patients living with limb-girdle to help us understand how we can become involved in the regulatory process in drug development. We have lots of experts that are going to be there with us. We have sessions all over 4 days. They're spread out very easy, and they start at noon every day. Registration is absolutely free to everyone and begins April 15th at internationallgmdconference.com. The first 300 registrants will receive a swag bag. If you're an international registrant, you will receive a chance to enter into a drawing, and we are giving away 10 $100 gift cards to Amazon, which is a great opportunity. So at this time, I'm going to introduce Dr. Alfano, and she can begin her slide deck. Dr. Alfano is a physical therapist with expertise in the care and evaluation of patients with neuromuscular disease, including outcome measure development, evaluation, and administration. She plays an integral role in planning and designing clinical trials and contributes to protocol development, outcome measure selection, statistical analysis, and interpretation for ongoing clinical trials. Dr. Alfano serves on an international consortium of experts that provide industry standard training and reliability for multi-site international trials. Her research goals focus on standardizing training in rare disease, as well as developing novel outcome tools to measure change in movement abilities while minimizing the burden of testing. And guys, remember, there is a live Q&A after Lindsay finishes her slide deck. So start asking questions now, and you can send them through the chat feature or in the Q&A feature through the webinar. So thank you, Dr. Alfano, and you can go ahead and begin your presentation.
Well, thank you very much for the opportunity to speak. And yes, I will second Catherine, your comment. Please ask questions. I'm always happy to hear what I didn't cover, or maybe what I didn't cover well enough, or what I didn't even think to cover. So let me know what questions you have. So outcome measures are my favorite topic to discuss. So I'm excited to have the opportunity to do that today. See if I can make my, there we go. So the first thing I kind of want to talk about is outcome measures as a whole, and kind of what the purpose is, why we have them, how we can use them and make them work for us. So I think when we hear the term outcome measure, we kind of think mostly about research and clinical trials, but I do want to stress that, it's really important to even have these utilized within clinical practice. So, we all want and assume that, kind of disease progression can follow this linear straight path, but we know it's a little bit more chaotic and crazy and scrambled egg-like. And so, we use outcome measures in our clinic to really help with the diagnostic process, to kind of inform expected trajectories and kind of talk about equipment prescription, using the outcomes to provide data-driven answers versus saying, this could happen in the next time period, insert that time period here, years, months, days. Having actual numbers gives us more information and more evidence to base our recommendations upon. Then similarly, we do use these outcomes for clinical trial readiness. So understanding what that disease trajectory should look like, what is a meaningful amount of change for a patient population. It may not matter if you change 0.1 seconds on a test, how much change is truly meaningful. And then really, outcome measures within the clinic and as part of natural history studies do build the team. So they get the patient community ready for clinical trials. You know what to expect if you're enrolled in a clinical trial because you've done these outcome measures. It also builds the kind of clinical trial infrastructure that we need for an international trial. We need doctors working with physical therapists, working with, social workers, working with study coordinators, working with that whole multidisciplinary team. And the more we practice at it with these natural history studies or having those multidisciplinary clinics, the more efficiently we can get trials moving and get products to market, which is really the goal. So the original question, what are outcome measures? So outcome measures are a vague term that describe almost everything. So, you may hear the term biomarker a lot. These are usually in reference to things that we can find in your blood. So different proteins, other compounds, sometimes in urine samples or saliva samples. These are, I'm not going to focus on biomarkers today, but that is a form of an outcome measure. We also use strength and function as outcomes for both clinic and clinical trials. There are other areas, so participation measures. So how are you able to participate in your home or your community or your work environment? So above, just a specific task, how integrated are you into your environment? And then there's also quality of life measures. So how happy are you with that level of strength or function or participation? So again, it's kind of this broad spectrum that encompasses hopefully all aspects of a person's life. I do like to start talks with this cartoon because I think the most common question I get from anyone is, well, what outcome measure should I use? That depends on a lot of factors. So just as all of these animals are very different and we would not ask them all to climb a tree, people are different, diseases are different, different cohorts are different, everything's different and variable. And so it's kind of a multi-factor decision-making process that we are thinking about when we're planning clinical trials or even selecting outcomes in clinic. So I like to think of it kind of like a funnel. We put everything that's available on the top, and then we go through our decision-making process and get rid of things. And eventually out pops the tool we want to use. But I want to get back to this. Why can't we just jump to the clinical trial question? Because I think we're all eager to get there. And hopefully, as I continue to talk, we'll see why there's this longer buildup to getting there. So as I said, planning for clinical trials is really key. So natural history studies, starting with natural history studies, is the best way to get to our answers as quickly as possible. As I alluded to at the beginning, we would love, as humans, I think we like to assume things follow this orderly path where we go from point A to point B, following this trajectory, but the reality is much more complicated. And so the more that we understand about what reality is, the more we can control some of those things that get us this spaghetti-looking picture to get closer to that linear path so that we can get to the answer we're looking for, which is, are you feeling better today than you did before? And is it because of something we did or not? So our goal is really to understand as much about a disease as possible. So understanding what causes that disease. Are there biomarkers, like I talked about before, that maybe are associated with disease severity or would give us some information about how likely a disease is to flare up or progress or have a change in function? We really want to know the effects of the disease on a person as well as that cohort or at a group level. So we can help, like I said, inform clinical care for a specific patient, but then also as a group say, is this group changing? What is affecting this group? What's important to this group? So we do that by measuring muscle strength and functional abilities, looking at other organ systems like cardiac function, pulmonary function, all the other body systems that could be impacted by a particular disorder. We really want to know how fast or slowly do we expect this condition to progress in whatever body system we're looking at over time. Because if we don't know that, then we don't know if something is working or not working. It's also helpful to really understand if a group of patients, so let's say limb-girdle muscular dystrophy, for example. So if you say, I have limb-girdle muscular dystrophy, do we expect every patient that walks through the door to follow that same path? No, because we have started evaluating and systematically evaluating patients with limb-girdle muscular dystrophies, we know that there are lots of different subtypes that have all kinds of different effects in different organ systems, and they may progress more quickly or less quickly. But without doing that kind of groundwork and starting to look into these specifics, we would be at kind of at the ground level and unable to see these more kind of smaller changes with treatments, et cetera. So ideally, we would really have these natural history studies completed before a compound exists, or if potential treatment options are coming down the path, we'd like to get clinical trials or these natural history studies started at the same time so that we're learning as we're studying a compound. Before is better. So taking natural history, how can we use this for our clinical care? So as I've said, we really want to understand how a condition should progress. So we can say, you are doing better than we expect, or something's different. You are progressing much more quickly. Let's figure out why. I think it's easy to say, you have a limb-girdle muscular dystrophy, your disease is supposed to progress. So see you again in a year. But often there can be changes in function and changes in your abilities that are not in any way caused by your limb-girdle muscular dystrophy. So maybe you had some sort of back injury that makes it really hard for you to do this particular movement or activity. That may be treatable and be not related to your limb-girdle muscular dystrophy. Similarly, you could have an ACL tear because you moved slightly the wrong way. That is treatable. And so the more that we understand this trajectory, we can really hone in on, changes or differences or other causes that could be impacting that trajectory over time. We really want to proactively plan for future needs so that, people don't wake up one morning and they're unable to do the important activities of daily living that they need. We want to make sure that people have the equipment and assistive devices required to stay as independent as they want to be. And then, people are trying things while there aren't any necessarily FDA approved disease modifying treatments right now for limb-girdle muscular dystrophies. That doesn't mean people aren't seeking treatment options. So having numbers associated with you at point A, maybe you have physical therapy treatment, or maybe you started a different supplement or some other sort of treatment opportunity. We can say, yes, your numbers are actually much better than they were the last time we saw you in March of 2021, or it's about the same. So if you feel like it's helping, it's not hurting, the numbers look good, but it gives us that evidence to really back up our recommendations. So back to this clinical trial. So using these natural history trials to inform our clinical trials or interventional trials, we really need to know what that typical disease looks like. The FDA doesn't know. You understand how your disease impacts you, but the FDA doesn't live in your shoes. So they need numbers to be able to say, okay, cohort 1 started at a 7, cohort 2 was at a 7.5. Here's where they ended up. Is that different? Do we believe that this is truly changing? and impacting this cohort. So they often don't consider a natural history study equal to a placebo group, because often, placebo groups are getting more regular follow-up and care. And so there's some thought that maybe that can impact outcomes. But the more information we have from natural history studies, usually the quicker trials can be. We can often have shorter trials, or that we can more rapidly enroll in those trials. The FDA and other regulators really want to understand the magnitude of the treatment effects. So they want to make sure that if a treatment, let's say numbers are different between the two groups, they want to know that that benefit truly outweighs the risks associated with any potential treatment. So the more that they understand, the easier it is to paint that picture or provide that narrative that yes, this treatment is working and is useful and should be approved. So we're finding the balance. That's the goal. So people are different. The same condition can be different across different people. All of the different sources of variability that occur, they all make it really hard to see that signal. So we want to find the signal through the noise. We want to get to the answer by understanding the disease progression and finding outcomes that are truly meaningful. They're impacted by the treatment, and we know what they should do in a certain period of time within a group. So just pick one already. No, that's what we're saying. Just pick one. Which one should we use? So these are some of the categories of considerations that we're thinking about when we're selecting an outcome. So it's the patient population. It's also the study, as well as the outcomes. So patients have their own variability. Pediatric outcomes may not be good for adult patients and vice versa. You're the study, so let's say this is a clinical trial and you were hoping to improve upper extremity strength, then it does no good to have a walking outcome. Similarly, outcome measures in and of themselves have all different sorts of properties. Psychometric properties is typically how we term that. The way that they're kind of set up, the way that they're structured, how sensitive they are to change, how reliable they are. So putting all of that together, we kind of go through that filtration process and we end up with our outcome of the trial. So to take that kind of theoretical information and make it a little bit more concrete. So thinking about different patient populations and applying a test that we're all familiar with. So I'm sure many people on the call have heard of the 6-minute walk test. It's a very common outcome. I like to use this as an example because it's an outcome that's existed for a while. And I think as humans, we like what's comfortable. So we often use what has existed for a while because it's existed for a while. But then that can also cause some issues. So the 6-minute walk test actually started as the 12-minute walk test back in the 1960s in the armed services. It was published in 1968. They finally standardized the administration of this 12-minute walk test, and then it was used for decades because everybody used it. So in the 1980s, Dr. Butland et al. Compared the 2-minute, the 6-minute, and the 12-minute walk test because they thought, we've been doing this 12-minute. Could we do a shorter test? And so what they determined was that shorter tests were easier, believe it or not. They're as reproducible and have less of a training effect than the longer test. But they also thought they discriminated slightly less well, which what they meant by those words were there was less variability in the total distance people could go. So maybe they couldn't really measure fatigue with it. So their scientific conclusion was the 6-minute walk test is in the middle. We should pick that. Seems like a sensible compromise. And so for then the next 20 to 30 years, everyone used the 6-minute walk test because it had been validated in this 1980 study. When I started in this role near the 2010s-ish, we started using the 6-minute walk test in every clinical trial that we were involved in. And what we started to see was an increased burden of testing. So patients were walking farther than they felt they needed to or wanted to. Many times patients walk at a specific speed, so I can measure that in a much shorter distance rather than having you walk for 6 minutes just to walk for 6 minutes. And we saw some challenges in a pediatric population. Kids don't necessarily understand minutes or time. So asking them to walk as far as they can in that 6-minute time period was a little bit problematic. So we decided to look into other outcomes. Do we truly need 6 minutes? Are shorter distances useful? And so in an adult cohort, what we found was if we looked at a 2-minute walk test and a 6-minute walk test, we got about the same results. So patients walked the same distance in 2 minutes as they did in the first 2 minutes of the 6-minute walk test. So we saw this nice straight line. You see a little dip here, but that's a meter or two. It's not really significant. So what we found is in patients with this inclusion body myositis, we didn't really need the full 6 minutes. We got the exact same information in those first 2 as we did across the entire 6-minute walk test. So we recommended a shorter test. Similarly, as I said, in a pediatric population, we thought time is really abstract. We all do better when we can see a finish line. So what if we have patients go as fast as they can to the finish line? So we use that same track, but instead of saying, okay, child who's 6 years old and doesn't understand what time is, go as fast as you can for as long as I tell you to. It's you do 2 laps of this track, down and back, as fast as you can. And when you're done, you are done. So they are motivated to get to that finish line. And so what we found is we had really high, what we call test-retest reliability. They were going almost the exact same time when we made them do it twice in the same day. People were concerned we were allowing these kids to walk or to run. They were concerned about falls, but we actually had less falls than we do in that timed walking test because they were motivated and focused and they didn't get distracted or, they weren't looking all around. And we were able to see some results that showed us that this test was measuring what we thought it was when we could see differences in treatment groups. So that was really exciting. So to kind of summarize that bit, I just want to say that, outcome measures can and will continue, I'm sure, to change over time. We continue to build on this great foundation of knowledge we have across neuromuscular diseases and other fields as well. And so we can pull that knowledge together. So it's not just that we're throwing out outcomes and moving on to new things. It's making sure that what we're using is the best for the given population that we're working with. Natural history studies really help us prepare for both clinical trials and provide better care within the clinic. And so, working together as this huge global team, I think we can continue to work to advance this field of outcomes. So quickly, I wanted to talk about some common outcomes you may hear when you are in clinic or when you are involved in a clinical trial, or you may have just heard in passing and wondered, what exactly is that? What are they talking about? I'm not going to go into specific details about all these and how they were validated and all that geeky stuff that I enjoy, but I'm happy to talk to anyone about any of that at any time. But more just kind of what is it? What do I do if someone asks me or if I'm supposed to do this test? So there's lots of ways to measure strength. So strength testing is something that you have all probably had at some point in life. Manual muscle testing happens frequently in the clinic environment to understand how different muscle groups are strong or less strong and give us kind of a better picture of how a certain disease may be affecting a person to come up with a diagnosis or look at kind of gross, larger changes over time. Handheld myometry is kind of a class where we use these handheld myometers, hence the name, where we hold them in our hand and we as a physical therapist would push against, a joint or a particular part of the body. You would push back and we could see how strong or how much force is generated in that particular area. There are also fixed systems, like as pictured on the right side of the slide here. And here, you have straps and force transducers. It's very sophisticated. Everything has to be parallel and perpendicular, but it's quite sensitive. And we can measure really small to really large amounts of strength. So as you can imagine, there are pros and cons to each of these. So this is kind of that decision-making table we would think about if we're trying to select an outcome for clinical use or for use in a clinical trial. Manual muscle testing, like I said, quick, easy, free. I can test a bunch of muscles, but I'm probably not going to use it for a clinical trial because it would take a large amount of strength to change from a 3 to a 4. Conversely, handheld dynamometry or myometry is kind of in the middle where we can still test a lot of muscle groups. It's sensitive to change because it's, I know exactly how many kilograms or pounds of force somebody is pushing. But it can be a little bit difficult because it does require you to sit still. So for childhood or pediatric conditions, it can be a little bit hard with keeping someone in a position and not squirming all around. And then, like I said, that fixed system or the quantitative muscle analysis is really sensitive to change. We can get, really fine-grained detail about strength throughout the body, but it is very time-consuming. It's expensive and large equipment, so it may not be feasible all the time. So here, I like to use, I don't want anyone to get bogged down in the numbers, but we use this graph at the top, this blue graph, to show kind of the range of strength you can find in different manual muscle testing groups. So if you look at kind of the middle two boxes there, a score of a 3-, which means somebody can move their limb through part of their range of motion. They may be able to generate 0.9 kilograms or about 1 pound of strength, or 2 pounds of strength, math, yes, all the way up to about 22 pounds of strength or 10 kilograms. And then you see that there's some overlap with patients who then would score a 4, meaning that they can move their limb through their full range of motion and they can take some resistance from the physical therapist or from the doctor without, that shoulder giving way or the knee giving way. They're generating between 4.9 to 18 kilograms. So there's overlap, and that's a really wide range. So if you start at 4.9 and have to get all the way up to 20 in a clinical trial, that is a lot of change and may not be feasible in 6 months or 3 months or however long the trial is. Similarly, function. This is my favorite area, so I will try to be brief. But the North Star assessment for limb-girdle type muscular dystrophies is an example of an ordinal scale that was developed specifically for limb-girdle muscular dystrophies. And here we rate patients as unable. They can partially do a task where they have achieved the task. It gives us a really nice kind of full picture of motor function. And it's got really nice reliability. We can detect change over 1 year, and it's a nicely constructed scale. Something shorter that you may have heard of is the timed up and go, where you get out of a chair, walk 3 meters, come back and sit down. It's a timed task. It's a very specific task, but it gives us some information about strength, function, as well as walking speed. There are also a variety of balance measures that we use in clinics where we look at fall risks. We can do kind of standardized, really challenging scales like the Brunig-Sastre scale, which has challenging balance items that I fail regularly. And then there's also more function-based balance tests. Like I said, the timed up and go has a bit of a balance component to it. Similarly, stepping up on steps without using support, all of these things give us more information about a person's function and balance. But even though I'm a physical therapist, I don't want to focus too much on motor, although that is my area of expertise. There are a lot of other systems of the body, so cardiac function, pulmonary function, other functions like swallowing, speech, cognition. You pick an activity, there's probably an outcome to measure it. So I don't want to focus too much on motor, although that's a lot of where my research would lie. And just because we can measure it doesn't mean we should or that we will. So is it meaningful to the patient? And let's say this is a clinical trial. Do we expect it to change? So a lot of times trials will focus on that low-hanging fruit. What do I think is the first thing that I'm going to be able to see change in this trial? That's what I want to measure. It does, sometimes they'll include other measures as, safety measures, but maybe we don't expect your cardiac function to change, but we want to know, or improve, but we want to know if it gets worse, because that means something's happening and that this treatment is affecting the heart in a way we wouldn't expect. And then, is the outcome sufficiently validated? That's another issue. So a lot of times, yes, this is an important and a meaningful thing that could change with treatment. But if we haven't studied it and we don't know what it should look like, we can't really throw it into a clinical trial, because we can't say it's gotten better than we would expect, or it's stayed the same when we would expect it to get worse. If we don't know that, we can't really use it. And then people also ask, you'll hear the words patient-reported outcomes or quality of life tools, and what are those? There are tons of them, so I'm not going to go into specific detail about all of them, but just know that there are a lot. And what it stands for is a patient-reported outcome. So this is really anything that a patient would respond or answer questions. So it's a chance to hear that patient voice in a standardized way versus just having a list of anecdotal things. Oh, it's easier for me to put my shirt on. Here we can say at this visit, this was the score on these specific questions. At this visit, here's how these things scored. We do want to find the balance between asking books worth of questions about daily life and not asking enough questions. So we realize that while there are tons of questions we could ask, we still try to focus on those things that we would expect to change and that are meaningful. There's also health indices, which ask questions about activities. So how easy it is to get around, take a bath, take a shower, use your arms, versus kind of quality of life measures, which are more about how you feel about this condition, how this is affecting your overall quality of your life. Remote testing, I think, is a positive of a pandemic, which has had a lot of negatives. I think before this pandemic started, everything you were required to do was in a clinic environment. The pandemic has forced us to adapt or get no data. And so I think there are a lot of validation efforts underway. I know at our hospital in particular, we're looking at, our tests that we typically complete within the clinic capable of being completed in the home environment in that standardized way that allows us to use them to inform clinical care and or clinical trials. The answer is yes, we've been able to do this successfully. Now, whether it's exactly the same as in the clinic, we are still gathering data to answer that question. But we have been able to do a lot of different outcomes in the home environment. Just know this slide depicts that some of the things we do for clinical purposes, we don't need as much standardization, but if we're using it for a clinical trial, we have to send a whole bunch of equipment into the home, which often isn't feasible, but is still possible. So if I haven't convinced you yet, how can you make outcomes work for you? So I wanted to show some examples of how we use these in our clinic environment and for clinical trials as well. So in an adult population, we looked at walking. We compared walking distance to different strength throughout the body. As we know, walking is not a one muscle group sort of task, but how do these different muscle groups interact? And what we found was that different muscle groups were contributing differently to the distance that patients were able to walk. But what we thought was more important, in a condition where you may not have a feeling of having much control, one thing that is somewhat controllable, though may not be easy, is weight. And so we found that for every 1 kilogram of weight that someone had, they would walk 1.2 meters less on this walking test. So again, not saying it's easy, but in that instance, we can say, look, because of this pandemic, we've all been less active. We see a 3 kilogram increase in your body weight. Let's work on this to see if we can make you feel less fatigued with, that activity or walking like you used to do with, your dog or your friends around the block. Similarly, we've got a video game that we developed at our institution where we tried to distract you from doing the hard tasks. So we have patients reaching and leaning in all areas of space. And so we can quantify the volume of space that people are able to access. And what we find are there's critical thresholds for when a task becomes really challenging. So if you can access X amount of space, you're doing well, you're probably not reporting any issues. Once you fall below this threshold, that's when you're going to really tell us that this task has become hard. And so what we can do is over time, track your abilities and say, hey, we have noticed there is this change. We're worried we're getting to this threshold. Let's see if there's something we can do to intervene. Do we need to get some sort of assistive device to make this task easier so that we can still maintain that independence proactively instead of having patients report they've lost the ability to do something. Similarly, we can use that 100-meter time test I talked about earlier. There are critical thresholds for when we see patients have more difficulty with walking or start to transition to wheelchair. As we know, wheelchairs and other equipment can be really challenging to access. So it's a lengthy, time-consuming process. So the earlier we start that process, the less it can impact a person's independence in their home and community. So to conclude, before we get to all of the great questions you guys have put in the Q&A, I hope I've convinced you that at the very high level, outcome measures can be useful for both clinic and research. I've stressed that natural history studies are really essential. We really need to understand as much as we can so that we can get to that answer, whatever that answer is, whether it's a clinical question or a clinical trial question, as quickly as possible. They do allow us to use patient input to design new tools. So please, please, please share your thoughts with your team, with everyone about what's working, what's not working, what we're missing, because we really do take that into consideration. We really want things to be meaningful. If it means something to a patient, it'll be more consistent, it'll be less variable, and it'll get us the answer we need. But our goal is to really be prepared, improving, maintaining as much independence as possible, and again, getting to those approved treatments as quickly as we can. So with that, I'll just say thanks and take any questions you guys have.
Thank you so much, Dr. Alfano. What a great job you did. We have a lot of questions that have been pre-submitted as well. So I want to start with a few of those, and then let's get to these live questions. And you kind of alluded to this already in your presentation, but one patient sent this in. What is considered a patient-reported outcome measure?
Yeah, so patient-reported outcomes, like I said, are anything that the patient is answering. So they come in all shapes and sizes and varieties. Sometimes we're asking about upper extremity abilities. Sometimes it's about fatigue. It's really a tool that we would use, a standardized tool that we would give to patients to have them give us feedback on how they're doing in that particular area.
Okay, thank you. Along that line, is that weighted equally to what is done in clinic for outcome measures?
Yeah, so that's a great question. So I would say as a physical therapist, yes. So I think it's just as important to hear, we always combine our clinical assessments with our patient-reported tools, because it's not just about what I think it's important. We want to hear the patient voice at every visit. And often we may see that maybe our clinic, the things I'm collecting have stayed stable or have changed in any case, and we'll see something different in that patient-reported measure. And so that gives us more information that we can then have that discussion with the patient. When you're thinking about clinical trials and regulators, they do put a lot of weight into patient-reported outcomes. They want them included in all clinical trials. Where it gets tricky is sometimes over time we see some noise in those patient-reported measures. So we do need to have the exact same sort of standardization and natural history studies in those patient-reported tools so that we know we've got the best, most reliable, least variable tool that we're giving to patients over time. So the quick answer is yes, they're very important and considered as such.
So I'm just going to share this really quickly. So for patients who are in natural history studies, when they're filling out their questionnaires often, you might not realize that that data is being used for a purpose. And I think a lot of times individuals will not necessarily think that that's weighted. They think it's just, data that's maybe taken but doesn't really matter. But it really does, because that natural history data could be compared to future treatment data in a clinical trial. Is that correct?
Absolutely. I mean, best case scenario is we have this natural history study, however long it is. We've done our battery of clinical testing. We've given you 4 hours worth of paperwork of questionnaires to fill out. And at the end of that, we find that one 10-item questionnaire was the best thing that we had throughout the assessment. So then it becomes, this is our clinical trial outcome. We will give you this questionnaire, your 10-item questionnaire, to tell us whether or not this drug works. We haven't quite gotten there yet, but wouldn't that be great? So yes, please, I would stress what you just said, Catherine. Take them as seriously as you take anything else, because if all we want is the most valid and reliable and sensitive to change tool that we can find.
Yeah, that's important. Why do natural history studies matter so much? A patient put that in. I guess what they're meaning is, there's so much emphasis on it. Why does it matter so much?
Yeah, I think we would all love to just jump right into the clinical trial. I mean, myself included. As soon as I hear that there's a new gene therapy or a new small molecule that's being developed, it's like, okay, well, let's get to it. Let's see if it works. But the let's see if it works is what's hard. So, there's a wide range of people. So we have people that, you could barely tell that they have a diagnosis. And then there's people where, yes, they're significantly impacted by this disease, and maybe they don't have great movement, or, they've got some significant heart and lung impact. So who are you going to test for your trial? It's hard to answer that unless you know which groups you expect to change or not change. Which outcomes should you use for each of those groups? If we are just guessing and we pick what we think, so let's say we say the 6-minute walk test has been used in 10 previous trials, and some of them were successful and some weren't. So let's just throw the 6-minute walk test in there. And then all the patients that enroll can only walk for 2 minutes or less, or maybe they can't walk. Well, now our treatment isn't going to look like it works because we picked this outcome measure that everybody's used. It wasn't studied, but no one changed because that test was too hard. Or similarly, you pick an upper extremity test that is looking at all these different arm and hand functions. You throw it in the clinical trial, everybody gets a perfect score. But now everybody can do cartwheels and jumping jacks, and they couldn't do those before, but you didn't measure that. So the more information we know about a population before we jump into a clinical trial, the more data-driven we can be, so that we don't waste all this time, money, and effort, and at the end get to the, that might have worked, but we didn't really measure the thing that we should have measured. That's the worst case scenario. So I think that's why I would stress participation in natural history studies. While it doesn't feel like it may be beneficial in the moment, is incredibly beneficial to moving clinical trials along as quickly as possible.
Okay, that's good. How do we change outcome measurements for trials for muscular dystrophy? A patient put this question in. I believe we need to have something for testing the diaphragm and or heart muscles. If my diaphragm or the muscles around my lungs were stronger, it would change my quality of life in a big way.
Absolutely. So I think there are two pieces to that. So one is it's important to have an outcome that we think we can impact. So some treatments may very well impact muscles around the lungs or those inspiratory muscles. Some may really have a dramatic impact on the heart. But if they don't, then they're not going to select that as an outcome. The other piece is having something that is validated, not to repeat myself, but validated and reliable. So there are outcomes for the heart, such as cardiac MRI or, echocardiograms, which can kind of measure the function of that heart muscle. So how much is it pumping? What are those values? And we know kind of critical cutoffs for those. But we also have to ensure that they are standardized in the way that they're administered. When people are scoring those, they're scoring them the same way, that all the different institutions have that same sort of MRI machine that can give the same valid results that are, if you're measuring something in Columbus, Ohio, it's exactly the same in Tokyo or in Newcastle in the UK. And if we can't do that, then even though it may be useful, it's not going to be included as an outcome for a clinical trial. But you can change outcomes for clinical trials by talking with your team about what's important. Because the more we know about what's meaningful, the more we can work to develop and standardize all those outcomes.
Yeah, I think that's where I noticed a few other questions around this topic. I think there's a topic here about advocacy and how patients can influence this, because if they think something's important to them, but they're not being heard, what are their options to get that out?
I have learned the more you say something, the more likely people are to hear it. So while you may not feel like, maybe a doctor at your clinic, you don't feel like this person is listening to your recommendations, you are welcome to talk to anyone and anyone in the field. So I get, emails and calls from people all the time saying, hey, have you thought about doing this? Or, I heard about this outcome you're doing. Have you considered tweaking it like this? And so, the squeaky wheel gets the grease, I guess. The more that you are communicating and advocating and talking to different groups, your voice is being heard. So even if you're not seeing immediate results, we are hearing that and continuing to try to drive these outcomes forward.
That's wonderful. All right, I'm going to get to some of our live questions because we are getting a lot of them. They're awesome. We had a question. Are there any general physical exercises that you recommend to individuals with limb-girdle? What can they do to slow progression or just maintain the muscle for a longer period of time? And are there any studies proving this?
Yeah. So that is a long question. That is a great question that I will attempt to as generally specifically answer as possible. So in general, there's not a worksheet that I could give that would work for every patient regarding exercise. What I can say is that every patient has to find that balance. So not moving is bad. Moving too much can probably be bad, and not moving, but overdoing it. If your body can't run a marathon and you force that body to run a marathon, that is not going to have good effects on your body. So it's finding that balance. What we recommend is having kind of a local team that you can utilize that can be kind of the experts in exercise, and you are the expert in your body. So the signs you look for exercise and kind of doing too much would be what we call that delayed onset muscle soreness. So a day or two later, you cannot move a limb because it hurts really bad or it gets sore and achy. That's a sign that you have worked those muscles to a point where they're kind of overly fatigued, breaking down, which we don't really like in muscular dystrophies because that breakdown and repair process doesn't function the same way as we would like. The other bit to consider is that what exercise would be interesting to Catherine might not be interesting to me. So you need to find what motivates you. So if I say, you need to walk 10 blocks a day and you hate walking, but if I said, a water-based exercise, you'd be all over it because you live in some lovely climate where you could have an outdoor pool and do that every day. It's really about what motivates you and is going to be something that you think is fun and isn't a task. So while that may seem simple, I think that's a hard thing. Everybody is looking for kind of that prescription, but it is finding the activity that allows you to be as active as possible without overdoing it, that you're not going to feel like it's a chore, because if it's a chore, you're not going to do it. And there is not a lot of great research in exercise across neuromuscular disease, just because it is kind of a hard thing to study because of all the variables I was talking about before. But there are some studies that show that doing exercise like cycling activities, these water-based activities, are safe and can be great for at least maintaining a level of strength and kind of aerobic function. And there has been some work that came out of the clinical outcome study funded by the Jane Foundation that showed that, sometimes too much exercise is correlated with a quicker disease progression. So we do want to keep that nice balance so that we're not overstressing the body.
Okay, thank you. Here's a question. How common is it for patient-reported measures to be used as primary endpoints in clinical trials?
Not overly common. But I'll say to date that has been because of a lot of the variability that we see in the results. And there have been, don't quote me on which one, but I know that there have been at least one approval that was based on FDA approval that was based on a patient-reported outcome. So it can be done. We just need to make sure that we have a validated and reliable tool that we're using.
If patients feel strongly about something, my concern, and I think I speak for, you know, not just myself, but for our community. Sometimes I think we feel that there could be a treatment that could work, but this is a slow progressing disease. So it's not as easy to, just see immediately. It could be that it might take a year or two to actually see that measure, especially if you're more progressed. I think it might be that it's more stabilized, like your
Mhm.
disease is more stabilized and it doesn't progress. And sometimes it seems as if we're looking for a Lazarus effect, like where, somebody's going to jump out of a wheelchair and start running. Where I think, from what I've heard from, I would say, I could conservatively say I've heard this from about 60 patients now. I would just be happy if you stabilized the disease and it didn't get worse. So
Mhm.
where's the balance for patients? This is my question on that issue of, I just want the disease stabilized so it doesn't progress any further, but how do you prove that?
Yeah. So what that takes is a really long placebo-controlled trial. And what's hard about those is signing up for a really long placebo-controlled trial. So often you could be, and let's say it takes 2 years for those groups to diverge. Patients would be signing up for a study where there's a 50-50 or 66-33%, whatever the randomization schedule is, chance that you would be in that placebo group for that amount of time. Which we, again, when we're planning trials, are trying to balance what we're asking of patients with that kind of benefit. So it's always that risk benefit. I agree completely that stabilizing function is a valid and important goal for a trial. I think the number of patients that are required to have statistical significance at some point is also an impediment for those sorts of things. So I think that's why trials tend to lean toward just below Lazarus effect, where there is a larger change because we can see it faster with fewer patients to then get to the approval sooner.
Totally understand it.
So it's that balance.
Yeah, and I think that's where a lot of patients, I think I see this question on our live chat about, can patients that are more progressed participate in clinical trials, and why is it that clinical trials tend to lean towards the younger age bracket? But it goes back to that issue, is that it's easier to see clinical benefit earlier on that in that disease, and then the hope is that when it gets approved, then all patients can receive it. And I think that would be the correct answer there, but I'm not the expert. What would you say about that?
Yeah, no, I agree completely. I think we tend to lean toward those patients where we would expect to see a larger change to get that trial over and done, that treatment approved, to then do safety studies in a different population to show, hey, this treatment is still safe, could be effective in this different group with the same disorder. So the goal is really to see that change as fast as possible, to get it out to as many people as quickly as possible. And there have been trials that start in patient populations that are, let's say, non-ambulatory or older populations. And sometimes there are other conditions that exist. So, maybe your heart has some early cardiomyopathy, or there's some lung involvement. If we do see an adverse event that happens during the trial, then the question is, did that treatment cause it? Or would this have happened with your disease? Is this expected? And so some of those questions researchers like to shy away from, to get, again, to that answer as quickly as possible, to then get the treatment out to everyone as quickly as possible. That doesn't mean it's right or fair or okay, but that's often the thought process that goes into it.
Sure. We have a question from Jason in the UK. He said, how do I sign up for trials? I am newly diagnosed genetically. I think he's saying he has limb-girdle 2L. I'm based in the UK. Do I need to travel? And is there funding? And that's a good question.
That's a good question. Yeah, so there are, well, the exciting thing is there are trials going on in limb-girdles right now for different subtypes. There are natural history trials going on. So there's the GRASP consortium that exists in the US, and there's also sites in Copenhagen and Newcastle upon Tyne in the UK. And so again, those are enrolling patients with 2L specifically, as well as other subtypes of limb-girdle muscular dystrophy. And so that's an opportunity to participate in that sort of natural history trial. And then there are some treatment options coming down closer to getting to patients across a variety of subtypes. So typically these experience sites like the Newcastle or the Nationwide Children's in Columbus tend to be sites for those. So once you're kind of established with those clinics, then you hear more updates and it's easier to kind of get involved.
Yeah.
Funding depends, but.
That's right. And, I wanted to share too, in terms of natural history studies, I know Sarepta has one going on right now. If you want more information on it, you could connect with advocacy at sarepta.com about those clinical trials for natural history. And there's also more clinical trials for other forms of limb-girdle as well that you can find on clinicaltrials.gov. And that's the United States site. And actually, you can go there and actually in the search box, put limb-girdle, and all the trials that have to do with limb-girdle will come up. And Speak Foundation periodically will actually advertise all the clinical trials. Well, we actually have our LGMD News magazine, and we'll actually put clinical trials in there just to get that in front of you so you know. And if you have any questions, you could always email us at contactus@thespeakfoundation.com, and we'll direct you to where you need to go to find out about more. Also, here's a great question. Boy, these are some great questions. Will the data of the natural history studies be accessible for all companies working on a therapy? Where's the data stored and who has access? Wow, that's a great question.
That is a great question. And the easy answer is it depends. So in general, usually, yes, that natural history data is available. It depends a lot on who is running the trial. And, if they're the data ownership rules, of course, there's all those sorts of things. At the very least, every group that runs a natural history study attempts to publish as much of that data as possible. And so even if that data doesn't get published in a usable form, often people can send a letter to the investigator, to the site, or to the sponsor and say, hey, we're really interested in this particular data set. Do you have this and will you share? So even if it's not published or it's not freely posted somewhere, that doesn't mean it's inaccessible. So don't be afraid to ask.
Oh, that's a very good question. So even if you don't see a published study, you could still request that data.
Mm-hmm. They may not share, but you can request it. So it never hurts to ask.
Let me ask you this about patient empowerment. When patients are enrolling in natural history studies, can they ask those questions?
Well, absolutely. They can ask about, where is this data going? Are you going to freely share it? I think the more questions that are asked, the more people will think about them. So sometimes, speaking as an investigator, I have my data set and I'm doing what I'm doing with my data set. But, when you've got this rich, huge natural history data set, there are so many questions you could ask, so many different ways you could analyze it. And so, I'm not going to think of all those and publish all of those specific things. So of course, I want collaborators to take chunks of this data and use it to get as many questions answered as possible.
Wow. I mean, honestly, Lindsay, this is such a great webinar. Like, I honestly could sit here all day and just listen to you. This is just such great questions that people are bringing up.
They're great questions.
They're wonderful. Okay, here's some questions. And I know these are specific studies that people are asking questions of, and I know that you probably can't answer some of this, but one question about the ribitol study, they wanted to know if it's been given to humans. I know they're seeing the 2I ribitol study, and that was a question there. Is that something that you can comment on?
I can comment vaguely. So since that is not my study and I'm not running it, I'll say that, companies, as they are dosing, whether they're moving from animal models to healthy humans, to humans that have known disorders, there are press releases that come out all the time that are publicly available. And so that's kind of you can often Google search those things and they just pop right up. I'd also say foundations like the Speak Foundation do a great job of kind of advertising all these results, or saying, hey, this group has just published this press release. This is the latest and greatest data. I do know that trials are moving quickly towards patients with limb-girdle muscular dystrophy. And so while they may not have started yet, I know that the goal is, again, to get there as quickly as possible. So fingers crossed.
Good. We have a question here. How long will it be before they have the gene for 2I to be ready to be implanted? And will a patient be able to get strong again and walk with exercise once they receive gene therapy? And I think this could be applicable to not just 2I, but to any of the gene therapies that are coming out.
Yeah, I think this is what's exciting is that we're getting these questions. So the fact that there's even the opportunity to ask about gene therapies and about what happens after gene therapy is an exciting place to be, because I know even that is just a hurdle that we hadn't even come close to crossing just a couple of years ago. So I unfortunately don't have specific timelines for any of the gene therapies. Again, I think companies are working as quickly as possible to get them into clinical trials. I think what we've seen in other diagnoses that have had gene therapies is that there are opportunities to gain strength. The muscles work differently. That there's different trajectories that appear after that dosing. Again, it's kind of individualized, so I won't be overly specific. But that's the next question we get to answer once we figure out is if the gene therapy is safe and effective. How effective is it? How effective can we make it?
Right.
So those are all on our radar.
It's still one of those questions that needs to be answered, right?
Yep, absolutely.
We have just a lot of comments too, just people asking questions about where Sarepta is located, and we can email you that question. If you email us at contact@thespeakfoundation.com, we can answer that. Europeans, question from a lady that lives in Romania. She wants to know, is she allowed to participate in clinical trials? Of course, I'm sure she can in her country, in her area, but are people allowed to come to the United States to participate? I think that might be a question she's got.
Yeah, absolutely. That's a great question. I will say, I'll answer not in the midst of a COVID pandemic. So speaking for our hospital in particular, we do a lot of first in human trials in gene therapy and other investigational compounds. And we have patients that come to our site from all over. So many different countries across the US, really almost all regions of the globe. I can think we've had patients come and enroll in our clinical trials. So yes, that is a possibility. There are a lot of hoops to jump through to do that. So, our hospital, for example, has an international group that actually helps ensure that all the appropriate, visas and insurance and all of those bits and pieces that I don't like to think about get taken care of, so that it makes that transition easy and possible. So it's nice to have, if you're going to those sorts of institutions that have those in place, make sure that you utilize those resources, because they can really be helpful.
Sure, absolutely. We have a question. If the study with 2E, I think a question here is, what is happening in 2E be used with all subtypes? I think this is a question regarding probably gene therapy. So I'm assuming that's what that means. If that particular gene therapy can be used for all subtypes.
Yeah. So gene therapies, if you're using, I do like how Sarepta kind of frames it, that they've got this engine. So there's a lot of pieces that go into that gene therapy. That specific gene is useful for a certain subtype of limb-girdle. So the 2E, for example, is beneficial for 2E, or they're hoping is beneficial for 2E. But what they can do with that sort of platform or engine is, take out that gene, put in the other gene that's useful for a different subtype, and then test that new engine in patients with that disorder. So the thought is that once it's deemed to be safe in one group, that's some foundational evidence to move the next groups along a little bit more quickly.
Sure. And we had a question, is there anyone that would be qualified to participate, I guess, in a natural history study? Or in particular, I know that each one has its particular parameters, but is there anything that would that disqualifies someone?
So for different trials, so natural history trials tend to have more open enrollment, so they have less restrictive inclusion and exclusion criteria. Really, you are often only excluded if you wouldn't be considered natural history, or if it's not safe for you to participate. So if you have, restrictions on movements or travel or whatever, and something to participate is not safe for you, then you'd be excluded from the trial. But in general, there are fewer enrollment restrictions in natural history studies. Clinical trials, where you're looking at an investigational compound, tend to have much more restrictive inclusion and exclusion criteria. And those are typically based on parameters that could impact safety. So, we want to ensure that the answers that we're getting, whether this treatment is safe, is truly because of the treatment. And like I said before, not something that was likely to happen as a result of the disease and just happened to occur in the clinical trial.
We had another question. Can the training for collecting outcome measures be standardized across sites? This individual said patients share with one another and find that our experiences are different across sites. So I guess she's saying in one site, it might be different what they experienced in a different site.
Yeah, so that's a great question. A lot of us around the globe are actually actively involved in standardizing training for clinical trials, so myself included. As I've been kind of harping on, we want to reduce as much variability as possible. So eventually, the goal is that, you could come to a Columbus, or you could go to somewhere in New York or in Romania, and it doesn't matter who you see in your clinic. It would be exactly this. It would be done in exactly the same way. It takes a little bit of time to get to that point. So that's why we, again, like these natural history trials, because they allow us to get more sites trained and reliable and doing everything in a completely standardized fashion, so that then when we do get to that clinical trial, all sites are, for the most part, operating as standardized as possible. And then over time, that branches out to more and more clinics.
Sure. Last question. If one of these therapies do provide a treatment that is effective, would insurance cover that?
That is another great question that I cannot answer definitively, but I will say there are gene therapies that have been approved in other diagnoses. So using spinal muscular atrophy as an example, the gene therapy for spinal muscular atrophy is approved by insurance companies. What's important is a lot of factors. So different insurance companies have different coverage cutoffs or criteria. And so in general, I can say, yes, these things are usually covered by insurance, but it does also depend on that treatment effect. Like I said before, the cost, the patient population, the labeling conditions, all of those sorts of things.
I think that's a great, question that someone asked, because, if the treatment
Absolutely.
becomes available, is the insurance company is going to stop it and halt and create a big complication for patients to try to get the treatment. And that's really important. That's where patients need to get involved in the
Absolutely.
advocacy process. And I will always say that, you might not think your voice matters, but it really does. And if you have opportunities to share your voice, I promise you, people want to hear from you. The FDA, there was a question, is the FDA easy to work with? And I'll ask you that. What do you think? Question that just popped up.
Sure. Well, it depends. Is anyone on the FDA listening? No, just kidding. No, so I think in general, I've had great interactions with the FDA. So anytime, I'm in a listening session, or we're having, pre-IND or IND meetings, they are incredibly helpful. I think what can be frustrating or more challenging is kind of like I'm doing right now. They don't necessarily give you specific answers because it all depends on what the data says. So, you know, they can give you kind of concrete recommendations or best practice guidelines or say, well, we'd consider looking at that. But they're not going to be able to say, yes, if you can give me X, Y, and Z, this drug will be approved because it depends on what happens. So that can be the kind of frustrating part is I just want an answer. Can you just say yes or no?
Yeah, that's right. You know, it's just so interesting. These topics are fascinating. I want to also just share as well that the patient community has something called a patient-focused drug development meeting next year that's in the planning with the FDA, where we get to share with the FDA as patients with limb-girdle what matters to us. And so there are multiple subtypes that are planning that and that are going to be involved in that. And so we'll have more information on that in months to come, but I wanted to throw that in there for patients interested in the regulatory process. I know, Dr. Alfano, you were so kind. You have actually shared that you would be willing to answer questions after the webinar.
Absolutely. Absolutely.
And so I want to tell everyone that if we didn't get to your questions, she is willing to answer them via email. So I want to give you an email so that you can send them to us, and we will basically send them over to Lindsay, and then she will reply. So if you have a question that you want to answer that wasn't answered today, I want to give you this email. It's contact us at thespeakfoundation.com. Just send us your questions. I know there were some questions that were more specific to certain patients, and I want to make sure that gets answered. I know there was one that was brought up that I didn't ask yet, but and I know that was by Kay Seifert. So if you want to send an email about that, we'll be happy to do that. And there was a couple of other ones that were really specific that I really think we could answer for you. But I want to thank you, Lindsay, for being with us today. And I'm just very thankful for everything you shared. You did a great job.
Oh, thank you. This is my favorite topic. So I usually start by saying I could talk about outcomes for at least 3 months, and I will try to talk about it in an hour. So please feel free to reach out if you guys have any questions. I'm always happy to answer.
Well, we're always looking for new webinar topics, and this one seems to be like extremely, extremely important. The community resources for Sarepta, I wanted to share with that is at limb-girdle.com. If you want to go there, you can actually find out about their community resources and join some of those there. I wanted to share that. So thank you to Sarepta Therapeutics for being our sponsor for these webinars. They're wonderful and great educational resources for patients, and I'm just very appreciative. And so I want to say thank you to everyone. This will actually be published on our platform and our website in 1 week. So you can go and check out all the other educational resources and webinars that we've already published professionally onto our platform. So thank you, everyone. And thank you, Dr. Alfano, for being with us today.